Good morning, everybody. Coming back to do a second post for the Fields Lab, let you all know what we're doing. This is my second time to do this, so I don't feel like it's very original. The first time, got a bunch of static. Audio quality in these things, that's a real thing to try to figure out. So I'm hoping this one is better. So this morning, what I was going to tell you about what we're doing, one aspect that I think most people can relate to, or you should be able to, and probably a lot of people have at least, even if you don't know, have heard about this or it may have affected your life. But talking about personalized or precision medicine, and that is the next stage or the next frontier in medicine. And what we try to do is we're trying to make it apply this principle to the brain, which is going to be more difficult because the brain's harder to get to. It's than other organs. It's more complex than other organs. It's wrapped in a blood-brain barrier that prevents cells and molecules from the periphery to getting into the brain for good reason, because it's delicate, as we all know. Uh, Things can fluctuate. You want to protect your head, right? There's a good reason for that. So one thing our lab is doing, as I said, is we're trying to implement novel personalized medicine strategies to prevent brain disease or help people take care of themselves. So let's start with sharing my screen, and already uh, let me get some things off of here. Working on a grant right now. But I've already started. I started with just a Google search. Personalized medicine. What does that mean? Um, Sorry, I've got folks messaging me. It is the morning time in the lab, so it's about to get busy, but I didn't want to go too long without posting something. So I haven't shared yet. I don't know why it's so difficult to figure out when I'm sharing, but now I'm sharing. So yeah, what I did was I just went onto Google, searched personalized medicine, and what do you see? What does AI say about personalized medicine? It uses an individual's unique genetic, environmental, and lifestyle information to guide disease prevention, diagnosis, and treatment, moving away from a one-size-fits-all approach, which is what we'd like, right? Because we've all seen those commercials, those pharmaceutical commercials, where you have a drug that treats restless leg syndrome or heartburn or something else. And at the end of the commercial, they have a list of other things, side effects that it does that don't look too great. And the way they determine those side effects is they tested this drug on 10,000 people, and maybe half of them, the drug did what it was supposed to do, prevented the heartburn, or restless leg syndrome, or whatever it is. But the other 50% of people, maybe half of them had worse heartburn, or they had headaches, or they had itchy toes, or whatever in the world could happen. And the point with personalized medicine is, instead of just saying, okay, this drug works on most people, so it's approved, we'll give it to everybody, and most people will benefit. Personalized medicine says, well, maybe we can take a closer look at the people, their genetics, their environment, or other things we think we can do in our lab, and identify before we give them the drug who would be receptive to the drug, who would be amenable to treatment by this drug, and it would improve their symptoms. So, as they say, it's an individualized approach. It's data driven. So that's what I'm talking about. They're talking about genomics, health records, how they've responded to other drugs. What do they do in their life? What is their diet like? And then, like I said, we have a different approach that I think it improves the resolution of the characterization. So it gives us more specific details, organ specific details on the patients, people you're trying to treat, targeted therapies. Um, That's right here. It enables the development and use of therapies that are specifically tailored to an individual's needs, potentially leading to more effective treatments and fewer side effects. Exactly. I think one disease uh, that has been using personalized medicine the longest is probably cancer, because you can sequence the genes and identify the mutations in the tumor cells that are leading to the cancer, and then target those genes. You've probably heard of this gene that predisposes for breast cancer. I think it's BRCA or BRCA1. Uh, but if women have this gene, it greatly increases the chance that they'll get breast cancer. And some women choose to go ahead and have mastectomies, just to prevent, they don't have to worry about it. There's other genes like that for the brain, like ApoE4 and TREM2 and APP. These are genes that if you have a certain mutant, a certain sequence of these genes, it predisposes for Alzheimer's disease. So you could take preventative measures right there to try to uh, stave off and prevent getting that disease. So in a picture here, this is just an image that kind of visualizes what we're talking about. You've got the whole population here. And the traditional way of getting medicine is if they have a headache or restless leg syndrome or achy back, you give them one treatment. And some people have good efficacy, some people won't feel it at all, some people have serious side effects. In a personalized approach, you can identify characteristics of these people to figure out which drugs they need, and they all have, not all, but a greater proportion of them has the good efficacy of the drug because the drug was tailored for them. Doesn't mean it'll be perfect, but it improves the outcomes of the treatment. Okay, so that's in general what personalized or precision medicine is. There's a lot of examples. I'm not going to go too far into them. I just hope you get the idea. And this is absolutely the frontier because, as I said, people are taking drugs that aren't working. I have somebody close to me who was having back problems and chronic back problems. And so they were trying all different ways to treat them. And they tried this one particular drug. Well, like a week later, after being on it, he had a stroke. And this was a young man. Scary, very scary. And one of the side effects of this medicine was that there's a low chance of having a stroke. Well, most people didn't have a stroke, but this particular person did. And if we could have known, if there was some way of knowing that this drug was bad for him, that would have prevented a huge amount of stress and a stroke. And it would have, so that's the reason for personalized medicine. And you can apply it to almost anything. Maybe you're one of these people that every drug you take has worked perfectly, but that's not most of us. So what is our lab trying to do about it? Um, I I would say that this is a paper we published that is starting to get into this area. I think I want to back up one more time. I want to make the point that traditionally, and this graphic shows it pretty well, people or clinicians use genetics or biomarker analysis or lifestyle analysis. So genetics, the sequence of your genome. Biomarker analysis, that's taking blood or cerebrospinal fluid, the fluid that your brain is floating in, bathing in, I should say, or lifestyle analysis. They take each of these three things, genetic, biomarker, or lifestyle analysis, um, and look for certain biomarkers that you can characterize an individual by. And in that way, you can decide what drugs they may be amenable to based on previous data from other people who have taken that drug that have that genetic analysis or have these certain biomarker profiles. Maybe they have more inflammatory proteins or more inflammatory lipids. And that gives you an idea of which drug to take because that characterizes their disease. We take a slightly different approach. And so what we do, as this says, is we take sera, which is just fluid, and it's the non-cellular fluid in the blood. In this case, we took it from people with HIV that had depression, and we took that sera and we actually added it to brain cells growing in a dish. That's something we do a lot of. We grow all different brain cells like neurons, astrocytes, microglia. We grow them in a dish to try to model the brain. Well, in this case, we took the sera from the people that had the problems, the cognitive problems, the depression problems, and we put it on the cells. And then we took metabolic readings of the cells to see if they had a relationship or they reflected in some way the diagnosis of the patient. And so in a graphical form, yeah, this is from the paper. So I think we had 40 people here that gave us sera. Half of them had clinical scores scores of clinical depression, and half of them did not. But it was on a continuous scale. I think it's like 0 to 40, and the lower number is no depression. The higher number goes up to severe depression, maybe 39. I can't remember. Um, we'll look at some data later. It's the BDI scale. Um, and so we get the sera from the people and we add it to this dish of astrocytes, and then we measure mitochondrial activity in the astrocytes. We look at it with our fancy microscope, and then we're able to quantify the activity of the astrocytes using this special dye that lights up in active mitochondria. And then we can characterize and quantify that activity by counting the spots, the intensity of the spots, the area of the spots, the number of spots, and then the total average intensity of all the spots. And when you take those numbers and you compare them to the depression scores and cognitive scores, I'll show you some both of that, you get an interesting result. In this figure, we're comparing it to cognitive scores. That is going to be 2. There we go. So on the y-axis, this is the vertical axis, you have the mitochondrial characterization. So how active they are. The higher up, the more active they are. And then on the x-axis, this horizontal axis, the numbers represent global, basically cognitive function. I'm keeping this very general. I am not a psychologist or a psychiatrist, but I work with some very good ones that allow us to do this kind of translational work. So basically what you see is the further you go out to the right on the horizontal axis, the better they perform cognitively. The better they can perform cognitively, the more mitochondrial activity that their sera induces in astrocytes in a dish, suggesting that whatever's in their blood is allowing for better mitochondrial activity in the brain. And that is a deeper characterization than just looking at genetic code of these people, right? And then there's some people that are performing cognitively, but they have, like this person here, they're performing fine cognitively, but they have lower mitochondrial activity. So maybe their mitochondrial activity in these cells is not so reflective, or it is not underlying their cognitive performance as much as these people. But these folks here, they have lower cognitive function and lower mitochondrial activity. If we could move their mitochondrial activity up, could we also move them out to this group here? And you can see this is not the strongest relationship. This is a pretty strong relationship. So actually, mitochondrial spot average intensity, I'm probably going too far into this, but the point is that's a pretty tight relationship there. Um, so if you have low cognitive function, these folks have the lowest mitochondrial spot average intensity. Um, so without going too far into the nitty gritty of these results, the point is there is a relationship in these cells, the mitochondrial activity in these cells after you treat them with the sera from the patients with their uh, cognitive function. which means, or what we think, is that in the dish, if we can try to reverse the mitochondrial activity in a dish with a drug or caloric restriction, something that mimics caloric restriction or ketogenic diet, then maybe we can identify what would actually help that person's brain. That's the whole idea. So that we could use cell models to get a better idea of the mechanisms that underlie their cognitive or any neurological dysfunction of the brain. And I just wanted to say that we do think we could even go further uh, by taking cells from these people and generating brain cell models that are specific to their brain, have the same genetic background as their brain, and then adding their sera and their CSF to better model what is going on in their brain and also better model how they would react to specific drugs. And we do have a patent pending on this. That is in this paper. I have to declare that. And I'm in the process of starting a company to get that going. I finally got approval from the university. They own my time, by the way. I hope they're okay with me making videos. So that's the idea. That's what we want to do. We think that we can use the cellular combination of our cell models in a dish for our models for brains in a dish with bodily fluids to better find mechanisms and therapeutic strategies for the patients. So I hope that like cleared up a little bit of what precision and personalized medicine is and how our lab is trying to improve it. So I will stop the share now and I will let you all have a wonderful Wednesday. Thanks for listening.